So this experience asked me to show some of the data that we have on the conservation of genomic sequence across different species, and what we know about it, and what we actually plan to do about it in DANIO-CODE. So one of the things that we have been working on for I don't know, more than a decade now is this non-coding elements of extreme conservation that exist across, first we were found across mammals, then across all vertebrates, and then separately pretty much in any metazoan lineage that we looked at. So these elements are actually known to actually occur in clusters on chromosomes, not randomly, so in clusters, and these clusters actually cluster around particular genes. And it has been shown that many of those highly conserved elements act as enhancers, and they all target these target genes. These target genes are often in regions of low gene density, but sometimes there are other genes that may actually contain these elements within their introns or beyond, but do not seem to be regulated by them. And we call those target versus bystander genes, and the whole structure we call the genomic regulatory blocks, because we think this entire region and this entire cluster of genes actually is of conserved elements defines actually a region from which this target gene listens to its regulatory input. And we have lots of evidence with it. I'm not going to show now, and many of you have already seen me talk about it anyway. But interesting thing is that we've had for a long time a way to visually locate regions of high density of non-coding conservation in the genome. One of those was our ANCORA browser, which dates from 2008. And so the idea is to find all the conserved non-coding elements and then and then display them as densities in sliding window view. And this sliding window density, the actual size of the window will depend on the actual size of the genome, because these clusters also shrink with the size of the genome. And so, for example, these are 5 megabase windows around 2 target genes. One is MACE2 and one is PAC6 in human. And you can see that pairwise comparison between human and mouse, and then human chicken, human and frog, and human and zebrafish. And you could see that this cluster can be 2-3 megabases wide. And 3 colors that you see in each, just 2 different thresholds for the elements. And interestingly, if you just zoom out in the higher chromosome, in this case chromosome 3 of human, each of these very highly conserved clusters of non-coding elements will lead you typically to a developmental transcription factor. And these are the genes that are most often found regulated by these elements. So some of the reference genomes that we have in ANCORA actually, of course, I showed you the human examples, but we also have several other species as reference. So basically you can browse either human genome or zebrafish genome or tetraodon or Drosophila or several others. And each of them has a pairwise comparison with all the species. Up to a couple of years ago, actually up to years ago, zebrafish wasn't the best species, the best genome to look at, because if you want to have conserved elements at different thresholds of conservation, what you want to have is a good range, a good scale of distances, evolutionary distances, from the most closely related ones to the most distant ones. And up until a year ago or so, zebrafish was an outgroup among teleosts. So all other species like tetraodon, medaka, fogu, and stickleback were actually more closely related to each other than they are to zebrafish. and the relatedness between them and zebrafish was about like between human and frog or similar. But in the past year, there was a Chinese group released one common carp genome assembly, which is also a cyprinid fish, which is fairly close. And there was also a grass carp, and we also have a European common carp assembly from Herman Spank lab here in is it Leiden? Then, of course, Tetraodon originally had this better scale, but now we have really some really close species to zebrafish, and this is just 10 megabase region. And you can see here is a stickleback, then Medaka, and spotted gar, which is outgroup to all of them, and then Medaka and Tetraodon, and then two very close related species, which is common carp and grass carp. And common carp, the problem is that it has another whole genome duplication. Grass carp hadn't, so I think this is better for comparison. But you can see, in addition to very highly conserved elements, you have less well-conserved clusters of the same kind that we see across mammals. We also have mammal-specific conserved clusters. And if you look at the orthologous genes, many of them have fish-specific conserved clusters, which just means that those clusters as a whole turn over around some genes very slowly and around some genes quickly. And we also have some functional dependence that we can associate with the rate of turnover. So one thing is actually, since this is those clusters, genomic clusters, are clusters of enhancers which are supposed to somehow interact with their target gene. We wanted to see what the relationship is within those regions and topologically associated domains derived from high C data or 4C data for individual loci. And we did it systematically by first developing a method for estimating the span of these clusters of high non-coding conservation. And now we have methods that predict across different thresholds and across different species fairly robustly the edges of these clusters of highly conserved non-coding elements. We just took all these, for example, one set from between human and chicken, and ordered them together, and we just, in the same heat map, we ordered them by size, and in the same heat map view, we showed completely independent topologically associating domain data. So essentially, what's red has surplus of interactions to the right, and what's blue has surplus of interactions to the left. And you can see that basically the boundaries of topologically associating domain correspond almost perfectly to the boundaries of these high non-coding conserved clusters. And this is, this is 8 megabase windows we're looking at in human. And if you look at it in Drosophila, now we're actually looking for conserved elements across Drosophila species, because there are none, almost none between Drosophila and vertebrates. We see the same thing, but the whole clusters have shrunk, but we see that it's practically the same phenomenon, and it's also targeting at the same genes. And the manuscript that actually is describing it is currently under review, but you already put it on bioRxiv, so you can read it. And so what the present future tracks in ANCORA will have H&E data sets and density tracks are already there. Yearly span predictions are getting uploaded, and so these are like proxies of this very stable developmental TADs across all the genomes and synteny blocks. And we are also going to add, since we always compare actual TADs, so we have TAD data, TAD span data, interaction data that we can also submit to DANIO-CODE, and we plan to do that. That's all.
I have a question that we discussed briefly, which is I just want to come closer.
Sure.
I can be heard. You can keep that.
Okay.
So could you please discuss briefly how could the ANCORA tracks be added to the track hub and used in independent browsers?
Right.
So
okay. So these tracks, if I actually show you, so these views are, so this is, we developed it a long time ago as a separate track plugin for gbrowse. This is for gbrowse, and this is just to show up to 3 different essentially bed file things, and there's nothing special about it, and any visualization that can show multiple density tracks can show these tracks. Okay, and of course, since there are so many of them, these kind of stadium-like plots that you showed are going to be very useful for zooming out and showing, things that are missing in different lineages, things that are doing it. And so this is, I think there isn't a big challenge in. These are all in standard formats. They're already uploadable to as individual tracks of either density or actual elements to any browser. But for like fancier visualizations, anything that can do the overlapping quantity tracks should be able to show it. There's nothing special about it.
Yeah. So again, I just come close to be heard, which is the question about organizing this, how to do it technically, because for now we only collected epigenomic data, transcriptional data, but none of these wiggle files for CPG island predictions and many other features which are inferred from other data sets, but would be extremely important to have together with the rest of the track hub. So I think we need to
it's an important, So the important, So the important thing is actually just for, to community, when you provide these tracks, that it's well documented how this was produced. So I mean, reproducibility for these tracks means that you know which software to run with which parameters to produce the same thing. Okay, and this is something I think. So this is not exactly what we currently have in ANCORA. We have been modifying our pipeline with years, but this is something for all the processing. We need to actually have a standard way to document how it's done.